Oh, okay, great. Can everybody take their seat? I know we're seeing people we haven't seen for a long time, and it's great to have you all here, but let's get started, and we're going to have some time afterwards for some socializing as well. So I just want to get us started here. I think I know everybody, but if not, I'm Michael Gropper. I came here in 1989 as an intern. I've been here ever since and had the privilege to train here in anesthesia and critical care and be on the faculty. I've been chair now for about 10 years. This is to celebrate John Severinghaus with our inaugural John Severinghaus Annual Lecture. John passed during the pandemic, and so we weren't really able to do something in a more timely fashion, and I didn't want to do it with any sort of restrictions or things like that so that we could really do a full celebration. John is so influential to everything that we do every day, and I try to make sure that the residents understand that when they're sending off a blood gas or giving opiates that are affecting respiration, or think about high altitude, or look at their pulse oximeter, that these are all things that were made possible by John. We have a connection to that legacy with just chatting with George Gregory right there, who was one of the people that came here and was profoundly influenced by John. We have lots of residents coming. I'm super excited. Come on in, have a seat. The back row might be taken, so you're in trouble. So I was a resident, and I have fond memories of John coming. He wasn't working clinically anymore, but he would come to the operating room all the time, primarily to fix the mass spec that ran into every single room with a little plastic tube and then ran into the ceiling. But also, I feel like I had the first view of the internet, because we could actually use those terminals to communicate with each other, and most importantly, late at night, to see when the isoflurane concentration was starting to go down in another room, and you were upset that your room wasn't finishing, but you could actually see the cases were finishing. So really a legendary figure. Some of you may have been here, and I was talking with his son. It must have been before the pandemic, 7 or 8 years ago. He came and gave really a remarkable lecture where he gave the talk that he had given because he was inducted into the Royal Society in London. It was really an amazing talk, particularly about the history of oxygen and Priestley and others, and a lot of the drama around that discovery of oxygen. When it came time to think about who our first lecturer would be, I think the logical choice was Phil Bickler, and he's going to be introduced by Judith Hellman. Judith is our vice chair for research and has also been here for about as long as I've been chair, longer than I've been chair. And she has done remarkable work supporting our research programs. So I'm not going to say anything more right now and introduce Judith Hellman, who will introduce our speaker. Olivia or Ed, can you switch us to the laptop here? Okay. Ooh, there we are.
All right. Hello, everyone. I will say at the outset that I did not get to meet John Severinghaus. I arrived here after he was no longer on faculty, or he was, but in a emeritus role. And I'm so sorry that I didn't, but I grew up on the East Coast and trained there, and he was a legend even on the East Coast. So, and I get to have the pleasure and honor of introducing Dr. Phil Bickler as the inaugural speaker for the John Severinghaus Annual Lecture. And I can think of no one that is more perfect to be the inaugural Severinghaus lecturer than Phil. He's another giant in the field of anesthesia research, hypoxia and pulse oximetry, just to name a few, which I'll name some more in a little bit. Dr. Bickler has spent the majority of his professional career in California and within the UC system. Although his impact and collaborations have gone and continue to go well beyond the confines of California into the far reaches of the United States and globally. He received his undergraduate degree at UC Riverside and his PhD in biology at UCLA, and then did a postdoc in physiology at UCSD and the Scripps Institution of Oceanography. That's a tongue twister. After that, he completed his MD and did a postdoc in physiology at UC San Diego. He then moved to UCSF, where he did another postdoc in the Cardiovascular Research Institute and Department of Anesthesia, notably under the supervision of John Severinghaus. After completing his third postdoc, Phil went on to do his internship at St. Mary's Medical Center and then his residency at the University of California in San Francisco. And he joined the faculty in 1991, so somewhere around the time when Michael Gropper was doing his residency here. Phil is a consummate physician and scientist. Throughout his career, he has literally done bench to bedside research, has been a leader in the research and both the research and clinical domains. He's the longstanding director of the UCSF Hypoxia Lab, which is the world's leading lab for studying hypoxia in humans. He also is the director of the, he's also has served as the chief of the Division of Neuroanesthesia since 2015. He's been a powerful mentor for numerous research trainees, including medical and graduate students, postdoctoral fellows, and faculty. And on a more global level, Dr. Bickler has gone on a number of missions to provide direct medical care in underserved areas of the world through the Operation Rainbow. Okay, so from the research standpoint, the breadth and diversity of Dr. Bickler's research is staggering. He's made important clinically relevant contributions in multiple areas, including in the basic translational levels to the understanding of mechanisms of hypoxia preconditioning of neurons, defining the roles of the endoplasmic reticulum and the unfolded protein response in neuronal responses to hypoxia, ion channel physiology, and basic understanding of gene expression as it pertains to the neurosciences. He's also made seminal discoveries and contributions to the understanding of human hypoxia physiology and in other diverse conditions, including, for instance, physiological problems associated with snake venoms. He's worked with multiple species, which I tried to show up here, including humans, obviously, rodents, jackrabbits, lizards, alligators, ducks, many species. And finally, he's played a key role in device testing, development, and assessment, and in addressing issues on the effects of skin pigment on pulse oximetry, which was a major topic of this morning's grand rounds. Phil's consistently, for all of his great work, had great research funding. He has published prolifically over 150 peer-reviewed papers. And I think with that as an introduction, I'm very much looking forward to Phil's presentation. Thank you.
Thank you, Judith. That was a very kind introduction. And thank you, Michael, for asking me to do this. It's really an honor. It's an honor for so many reasons. First of all, it's an honor because I love John so much. And I know there's many of you in the room who knew him and were so deeply influenced. And the ways in which he influenced my life and indirectly what we do every day in the operating room is something I want to share with you over the next hour. So my topic today is hypoxia, and I know John would approve. This is one of his abiding interests that guided so much of his contributions over so many years. So John invented modern blood gas analysis by combining electrodes for carbon dioxide, oxygen, and pH into what we call a blood gas analyzer. John died when he was 99, 2 years ago. John had so many firsts. He was the first to measure the uptake of inhaled anesthetics in the lungs, measure end-tidal gases in the operating room, and was a major figure in understanding the control of breathing and adaptation to high altitude in humans. John truly was among the most important thought leaders in anesthesiology in the last century.
So John was born the same year that J.S. Haldane published this book. It was called Respiration. That was in 1922. That book was incredibly influential in establishing a mechanistic basis for respiratory physiology. And I'll keep it up here. You can take a look at it. This isn't a copy signed by John. I wish I'd had him sign it at some point as just a memorial to the foundation that I knew he had and the debt that he owed Haldane and others. So John, more than anyone since, wove the science of respiration together with the art of medicine. That union enabled John's work to touch more lives than anyone our field has ever produced. He truly was anesthesiology's Leonardo da Vinci. John attended Haverford College in Pennsylvania and started medical school in Wisconsin, where his father was a professor. He completed medical school at Columbia, then started a residency at Penn, finishing at Iowa after an interruption where he was director of anesthesia research at the National Institutes of Health. I mean, it's hard to keep track of all this. I mean, his energy was downright frightening. As a medical student, he worked with giants like Julius Comro, who convinced him and Bill Hamilton to come to San Francisco to create our department and the Cardiovascular Research Institute. His work was foundational to the scientific basis of our specialty, and today I will be sharing stories about the most impactful of those to anesthesiology and also to me personally, really. Truly, John is the reason I went into medicine. He's the reason I went to medical school and became an anesthesiologist. I remember very clearly as a first-year graduate student in biology, discovering John's work on acid-base physiology. And I read the preface that John wrote to John Nunn's book called Applied Respiration Physiology. And I'll share that later. But after reading that preface, I knew what I had to do with my life. It was that impactful. You know, John's work, though, is so much deeper than the publications he wrote and the inventions he made. His legacy, you know, as I reflect on it over my career, was really in the generous spirit that he gave to so many other people that he trained. He trained people that went on to become major contributors in our field and major contributors outside our field, indeed in all of respiratory physiology. You know, and later in life, John continued his generosity, teaching our third-year residents about high-altitude physiology. And here's a couple of pictures that I'll share of John and the students that he loved teaching so well. John's greatest contribution indeed was the development of blood gas analysis. And these contributions have reached globally and affected the lives of millions of people. So Severinghaus's contribution really was to devise a carbon dioxide electrode that actually worked. And his seminal insight was that by adding bicarbonate to the electrolyte solution in a pH electrode, you could make it exquisitely sensitive to carbon dioxide. So if you combine a membrane that's permeable to carbon dioxide and you have a pH electrode, and the electrolyte contains bicarbonate, you force carbon dioxide into producing more protons that are detected by the pH electrode. And that was the basis for what's called the Stowe-Severinghaus CO2 electrode. And that is the same electrode that we use today to send a blood gas from the operating room to the clinical labs to measure our patient's CO2. It is essentially identical. It's been that durable. Pretty amazing. Well, the invention of blood gas analysis and creating blood gas analyzers influenced so many people. And I know you're here. Where did you go? George's work meant so much and continues to mean so much to all of us. And I think it's fair to say that George's care of fragile neonates was enriched so much by actually being able to measure what was going on in the arterial blood of these kids by being able to do a blood gas analysis. He could, yeah.
He started Lord, I think to carry you, and that's the one we had.
That's amazing. There's a copy of that machine that's in the Smithsonian. That's how impactful this was to medicine, to all of medicine. And and I think it's fair to say that the whole field of critical care medicine was propelled forward by being able to measure blood gases. I mean, think about it. That's just an incredible contribution right there. If he would have done nothing else for the rest of his career, he would still be just exceptionally revered. But John didn't stop there. So he really was innovating in all areas of patient monitoring. At the left is a Perkin-Elmer mass spectrometer. So this device, about the size of a washing machine, that big. And we had several of these in the Moffitt operating room when I was a resident. We didn't have standalone end-tidal gas monitors like we have now that all our residents are familiar with. We had only 2 of these things, and they were connected to each operating room by thin plastic tubing that went up in the ceiling. And via a terminal, you could command the mass spectrometer to open a valve and sample gas from your operating room. That way you'd be able to know whether you had put the breathing tube in the patient's trachea when you got carbon dioxide back, or maybe you put it in the esophagus and you didn't see CO2. But the cool thing about the terminals was, as Dr. Gropper said, is kind of the forebearer of the internet. We could communicate with each other. We could spy on each other. This was in like the 1970s and 1980s. I mean, amazing technology. And UCSF was the only place that had this. And it was because of John. Just, I think that's remarkable too. So when John was a medical student, he devised a way to measure anesthetic uptake in the lung. He realized that if you have a closed system with a patient breathing in it, if you absorb the carbon dioxide, you can meter in oxygen to keep the volume constant, and that would give you your subjects or patients' oxygen consumption. And by metering in nitrous oxide to keep the system balanced and equivolemic, you could measure nitrous oxide uptake in the lung. That was revolutionary. You could actually start to do pharmacokinetics of inhaled anesthetics. This guy right here almost owes his career to that innovation. That's Ted Eager, who pioneered the uptake and distribution of the inhaled anesthetics that we use every day. When Ted was getting on in years, one day he told me that the concept of MAC was basically John's. It was something that he suggested to him. So the minimal alveolar concentration is our universal standard for measuring anesthetic potency. John had the basic kernel of that idea. Ted Eager, Larry Seidman, and Brandstatter worked together to devise the rest of it. And we all know how influential that was. John had his hand in it. John was really interested in how our breathing is regulated. And he contributed so much to the understanding of both how oxygen is sensed by the carotid bodies and how carbon dioxide is sensed in the brain. So John knew from experiments with Comro that animals still had a drive to breathe if you denervated the carotid body. There was carbon dioxide sensitivity somewhere else in the body, but nobody knew for sure. A logical place to look, they thought, was in the brain. And with his encouragement, Bob Mitchell and others in the CVRI identified areas on the surface of the ventral medulla in cats that were responsible for chemoreception of carbon dioxide and pH. And so a more complete picture began to emerge of how breathing is regulated. This conception became very important to us. It was actually John Feiner and I, along with Mike Lipnick, we wrote a paper on the control of breathing that was relevant to patients with COVID. So we published a paper early in the COVID pandemic, which explained how the carbon dioxide sensing and low oxygen sensing was integrated to produce increases in breathing, and how there was such an intricate balance, and how these derangements could play out in patients with COVID. And we also explained how some patients lacked very much drive to breathe, the so-called silent hypoxemia of COVID. That all fit perfectly into the understandings that John gave us. So I want to talk more about hypoxia, and not only John's exploration of hypoxia, but mine as well. And a great place to start is in 1875, when French balloonists were trying to outdo the British. There was an international competition to see who could ascend the highest in balloons filled with hydrogen. And the French balloonists ascended in a balloon called the Zenith. And the French had a secret weapon, a guy named Paul Bert, who understood the effects of barometric pressure on the availability of oxygen in the atmosphere. So Paul Bert gave the balloonists these goatskin bags filled with oxygen, or about 60% oxygen. And he told them, when you start feeling funny, you can start breathing the oxygen and you'll be fine and you can keep going in the balloon. But I think they either got excited or confused and they didn't do it. And it was a disaster. 2 of the 3 died. The third barely had the wherewithal to record that they had ascended to 8,500 meters and to descend. They had a crash landing. And I don't know what happened to the balloon competition, but that was definitely a setback. So Paul Bert was a French physiologist who wrote this book called Les Pressions Barométriques. And this became the foundation for understanding what happens to humans when they ascend to high altitude, because the barometric pressure goes down, the fraction of oxygen stays the same, the oxygen partial pressure decreases. So if you're at the summit of Mount Everest, the inspired oxygen level is going to be only 53 millimeters of mercury. Think about a patient of yours who has an arterial PO2 of 53. You'd be hitting the code blue button probably. It's a real problem. So that understanding led to the assumption that high altitude climbers had to take oxygen with them. And in 1953, the British finally summited Everest. This is Sir Edmund Hillary and Tenzing Norgay, each with backpacks containing cylinders of oxygen. And they made it to the top of Everest, the first humans to do so. And it was firmly fixed in everyone's mind that without oxygen, Everest would have been impossible. However, in 1984, a guy named Reinhold Messner, a very accomplished alpinist from Austria, set foot on the summit of Mount Everest without using supplemental oxygen. Absolutely astounding. I don't know how many of you are climbers, but this is just like an amazing feat. And it ranks up there with like Alex Honnold and Tommy Caldwell, these guys who free solo, in Yosemite, all these crazy ascents. So how did Messner do this? Some of the background for understanding how he might have been able to do it actually came from John West, who I worked with after I did my PhD and went to UC San Diego. I worked with Fred White, a guy who was a comparative physiologist at Scripps, but they had very intimate relationships with the West lab. I often regretted not finishing my PhD a year earlier because I might have gotten to go to Mount Everest with the American Medical Expedition. So what West did, he sent one of his critical care fellows. This guy's Chris Pizzo. He's a pulmonologist, critical care fellow from UC San Diego, who is also a climber. And West's lab created this device so they could sample a sample of end-tidal gas. So Chris took off his oxygen, which he used to climb Everest with, breathed ambient air for 10 minutes, and then exhaled into this device. He squeezed the clamp, and the gas from his expiration was trapped in a small cylinder. The cylinder was returned to San Diego, where it was analyzed for CO2, and the PCO2 was 7.5 millimeters of mercury. That must have meant that Chris's arterial pH was like 7.8. Couldn't have been anything else. I mean, it was just, I'm pretty unbelievable. So how is this even possible? So here's a situation. So there's some residents here. You guys know that me and guys like John Feiner too are very fond of asking you to derive the alveolar gas equation. When you have a patient whose gas exchange is all screwed up, it's like, okay, what should the patient's arterial partial pressure of oxygen be? And you can use that equation to kind of figure out some problems related to climbing Mount Everest. So if you input the atmospheric pressure, assume an RQ of 0.8, and use a PCO2 of 20, which we know maybe is a little high, that leads you to an alveolar oxygen of 18 millimeters of mercury. 18. Let's say that's the PO2 in your lung, and you assume that you have no gas exchange gradient between the alveoli and the arterial blood. So then you can use the oxygen hemoglobin dissociation curve to figure out what your saturation would be. So with that assumption and Bohr shift to the left with extreme alkalosis, you derive a saturation of about 55%. Pretty amazing. So the guy in the inset is part of our story today too, because he was part of John's life. This is a climber named Tom Hornbein. And some of you probably knew Tom as the chair of anesthesiology for a number of decades at the University of Washington. Just one of the most delightful human beings you would ever meet. Tom was an amazing alpinist, and he pulled off one of 20th century mountaineering's greatest feat. He successfully climbed the west ridge of Everest, surviving an overnight bivouac at extreme altitude. It's staggering that Tom and his co-climber even survived. But he went on to become chair of anesthesia at the University of Washington. He also was a research fellow here at UCSF with Ted Eager and Severinghaus. Just remarkable. But in 2009, a group of British climbers got the first blood gas measurements from near the summit of Mount Everest. This is Michael Grocott and his team. And their arterial samples, they couldn't quite do it at the summit. They did it a couple hundred meters below the summit. But the data they got was pretty remarkable. The arterial PO2 was 24, PCO2 was 13, pH 7.6, and the saturation was about 54%. Can you imagine being a sat of 54? John and I have been at saturations of 54 because we've done it to ourselves in the hypoxia lab. But it is kind of uncomfortable. So closer to home, there's a place where faculty at the University of California have studied the effects of high altitude for a long time. This is at the White Mountains. So you know where the Sierras are. If you go across the Sierras into the Owens Valley, there's a big range of mountains beyond. In fact, White Mountain Peak, the summit, is a 14,000 footer. It's 14,250 or 246 or something like that. But the university has a lab at the summit and the Barcroft lab at 3,800 meters. So that's plenty high to do some serious altitude physiology. And I see Jeff and Danielle in the room. They've, in recent years, have done a number of expeditions to White Mountain to understand human adaptation to low oxygen environments, and also to see if they can do something about the problem of acute mountain sickness. But White Mountain was a favorite of John's, and his lab would do expeditions there just about every year. And I had the pleasure of going on a number of them. But White Mountain's high enough. It's above the elevation of Leadville, Colorado, and some of the highest inhabited places on the planet. When you first arrive at White Mountain, your SAT's like 85%. So you're ready for ICU admission when you step out of the car. So John was very fond of taking almost his entire lab up to White Mountain. So that meant blood gas analyzers. That meant the mass spec. we would put the mass spec in the back of a truck and take it up. It was an all-day drive to White Mountain over a really bad road. We'd take it up there, and of course, everything we'd take broke down on the way up. We spend the next week repairing it. John was a master mechanic. So some of the experiments that he did were just pretty unbelievable. So John was interested in how breathing is controlled at high altitude. He was interested in cerebrospinal fluid pH and how it was adjusted and how it contributes to increase in ventilation that gradually plays out over time. So I think it seemed obvious to John that what he ought to do is put a needle in somebody's CSF and then connect it directly via a tube into one of his blood gas analyzers. Makes sense. I mean, why do you need a syringe? Just like put it right in. I'm not sure who this poor chap is or what kind of headache he had afterwards, but I bet it was epic. So back to Tom Hornbein. So John was really interested in how brain blood flow is regulated and during high altitude hypoxia. And this is Tom Hornbein, the Everest climber. I think this is Larry Seidman. And of course, here's John wearing a necktie and a nice white coat, clean white shirt, supervising some horrendous experiment on Tom. There's a jugular bulb catheter. They're sampling cerebral mixed venous blood. And I'm not sure if this is a venous line or maybe that's a brachial arterial line. And then God knows what Tom was inhaling, probably something that would enable them to estimate the blood flow to his brain. Okay, and there's Tom again. So with that as a foundation, I want to talk a little bit about what I've done in my career. And I don't have time to go into everything, but I just feel so grateful that I've had the opportunity to study so many different systems and at so many different levels of biological organization. And if any of you want to ever hear about this, just let me know, because I would talk your ear off. But you know, in the course of my career, I've had the opportunity to study adaptations in humans, developing mammals, hibernators, diving marine mammals, and turtles. These have provided incredible insights into how systems can adapt to limited oxygen availability. You know, humans can extend their adaptiveness by a factor of 2 or 3-fold. In developing mammals, it's up to 10-fold. Same with hibernation. Diving marine mammals can hold their breath for 90 minutes. Pretty amazing. And most remarkable of all, turtles have a 10,000-fold greater tolerance of anoxia. After anoxia, their blood is basically black. Their heart's still beating. They can survive lack of oxygen for 4 to 6 months. I mean, just amazing. Taking it back to humans, I wanted to relay some of the messages that I think are really important, kind of a distillation of a lot that we've learned. You know, when we're in the operating room and we have some kind of a hypoxic event, we are very concerned about injury to our patients, as we should be. But what we've learned from our studies in all these years is that hypoxia is very well tolerated unless there's a failure of the circulation. This is a really key point that's missed on a lot of clinicians. Cardiovascular compensation in humans can be very robust and quite prolonged. And it isn't until saturation drops to levels that are around 50% that consciousness even starts to fluctuate. And when cardiac arrest finally occurs, you get bradycardia, then cardiac arrest, the oxygen levels are extremely low. Actually, they're probably close to zero. There's good evidence that the heart continues to beat when the PO2 is even less than 10 millimeters of mercury. But of course, if you're on that trajectory, the outcome is probably going to be quite adverse. So this business about loss of consciousness and when it happens, this was a very important topic early in World War II, because the militaries were producing ever more capable fighter aircraft that could ascend to very high altitudes. And pilots were losing consciousness, they were losing planes, they were losing their life. So Glenn Millikan, an American physiologist, was tasked with figuring out how to detect this. And he knew that blood changes color when it binds or releases oxygen. And he built a device. It was an ear oximeter. It had a light bulb in it, basically an incandescent light bulb and a couple of filters, and then crude photodetectors. So this would go on your ear as part of your pilot's helmet. And supposedly it helped pilots avoid losing consciousness at high altitude. Well, Hewlett-Packard got in the game, and they had a really interesting ear oximeter as well. I think people in the days of oximetry and oxygen measurements figured that the ear is kind of an obvious place to do it because the ear is thin, and that just seemed that was the assumption that the field went along with. So Hewlett-Packard's ear oximeter was an innovation because it had an inflatable cuff in it, and it would press on the ear and squeeze out all the blood. So it had a way of self-zeroing itself. And the Hewlett-Packard instrument had multiple wavelengths of light in it, and it was actually a pretty good oximeter. It's pretty clunky. This thing is really big. And it was used to a limited extent clinically. I don't know if we ever had one here. Severinghaus had one in the lab. I'm not sure what happened to it. Severinghaus developed ways of measuring oxygen in patients too. And he combined an oxygen electrode with his CO2 electrode into one unit in what's called a transcutaneous electrode, and had a little heater in it so that the blood flow would increase under the sensor, and it would essentially arterialize the blood in the skin. So Severinghaus thought that this would be a real innovation, but something got in his way. And what got in his way was the innovation of pulse oximetry. And this was done by a Japanese engineer at a company called Nihon Kohden early in the 1970s. Oyagi's prototype pulse oximeter is here with a kind of a cumbersome looking ear attachment. And this is John with Takuya Oyagi sometime in the 1970s. And the advent of pulse oximetry was a major event in clinical monitoring, and it was associated eventually with a 90% drop in mortality from anesthesia. Whether that's causative is debated, is certainly associated with it. So Oyagi's innovation was to use the pulse as part of the signal that was used to detect oxygen changes in the blood. So it's useful to think of optical components as being a static one, what's called the DC or steady component, and a pulsatile one or AC. So shining light through the ear or a finger or other tissues had both a DC signal and an AC signal to them. So this concept of using the pulse as a signal just spawned an enormous range of oximetry products. From Nihon Kohden's first prototype in 1972, in the early 1980s, Nellcor had this oximeter. I think some of our faculty probably recognize that one. It's about this wide, about this big, probably weighed about 75 pounds. I remember as a medical student in San Diego, we had a few of these things in the recovery room. And Larry Seidman was one of my attendings when I was in San Diego. I remember walking through the recovery room with him, and he would say, "This pulse oximeter is great. Before we got this thing, I couldn't tell if half the patients in here were dead or alive." And I quote. But you know, there's now rings that are pulse oximeters. There's smartphone oximeters, pretty gorgeous wristwatches, and of course the ubiquitous fingertip oximeters. All right, so what do pulse oximeters see? How do they detect these changes in blood oxygen? What pulse oximeters see is a quantity called the functional saturation. The functional saturation is oxyhemoglobin divided by the concentration of oxy and deoxyhemoglobin, just the hemoglobin that's available. This is in distinction to fractional saturation, which accounts for all the different species of hemoglobin, including carboxyhemoglobin, methemoglobin, and sometimes sulfhemoglobin. So pulse oximeters see this part of it. They totally ignore this. They may be interfered with by those things, but they don't detect it with just 2 wavelengths of light. So how do pulse oximeters derive this? Well, when the color changes, it produces differential absorption at a red wavelength and an infrared wavelength of light. And then the AC and DC components that I described earlier of each of these wavelengths of light is used to create a dimensionless number called R. That number R is related to saturation. So how do you get from R to saturation? Anybody know? There's only one way. It's this. You come to the hypoxia lab. Severinghaus realized that this was going to be required to produce accurate pulse oximeters. He published the first work on pulse oximeter accuracy back in the early 1980s when their pulse oximeters were just entering clinical use. That was around the time that I joined his lab. And we were really into self-experimentation. And if any of you want to look at my radial artery areas, I've got probably more dots from scars from having arterial lines than you can count. So pulse oximeters have to be calibrated in human beings. That's still true. No one's ever been able to develop a pulse oximeter that is independent of validation in humans. Pretty remarkable. So we have, over the years, I think you can fairly say that we're the world's leading center for this work. So we see just about, virtually all the manufacturers of pulse oximeters have been through our lab at some point or another. And I want to thank all the people that have helped us with this over the years. We have a lot of faculty who volunteer their time, and some in the audience who've been subjects. Greg, how many times did we desaturate you? You contributed in a small way to accurate pulse oximeter performance.
Good, Greg.
So here's a subject with an arterial line and a bunch of pulse oximeters on her. She's breathing a hypoxic gas mixture. Okay, so just some basics of how you assess the clinical accuracy of a pulse oximeter. So we are focused on the difference in what a pulse oximeter reads and what the saturation is in an arterial blood sample. That's the fundamental comparison that we're making, because with an arterial blood sample, you can put it into a device called a co-oximeter, and it will accurately measure the saturation. So that's our gold standard. So our reference for all these studies is what the pulse oximeter is reading, right or wrong, and what we see in arterial blood. And that's used to derive some various metrics. And one is called the ARMS, and that's just the difference between the measured and the actual, that pulse oximeter reading and the hemoximeter. You square that, you sum it, you divide it by n, you take the square root of it. That's this ARMS statistic, and that's essentially the precision of the measurement. And the FDA relies on this when they clear a pulse oximeter for clinical use. And the FDA and the International Standards Organization say that you've got to be less than 3% to be a clinically accurate pulse oximeter. Okay, so this bias can be in various directions. So if a pulse oximeter has a positive bias, that means it consistently reads too high, or it overestimates saturation. When your oxygen level is really low, it says that your oxygen level is okay. So that's a positive bias. A negative bias means the pulse oximeter is underestimating your saturation. So these are really important metrics. Early in the days of pulse oximetry, John said, I've noticed that our subjects who have darkly pigmented skin tend to read too high on most pulse oximeters. And John Feiner and I decided that we were going to study this in detail. So we recruited very light-skinned people and very dark-skinned people. We put arterial lines in and we desaturated them. And in 2005, we published these data showing that particularly at low oxygen levels, clinically used pulse oximeters, the ones we were using in that day, were reading too high. So they were missing the diagnosis of hypoxemia. We thought that was going to be a high impact paper, but that's what we got. No one seemed to, this was an inconvenient truth. In our paper, John and I even said, well, pulse oximeter manufacturers just need to fix this. So there's a correction. If you have a patient with dark skin, you use the correction. You flip a switch. It's not that hard. No one wanted to do this. So this idea kind of lay dormant until COVID came along. Suddenly, a lot of people were hypoxic, and people in communities with darkly pigmented skin were particularly impacted. And Michael Schoeding and his colleagues at the University of Michigan published a letter in the New England Journal showing that occult hypoxemia, or the misdiagnosis of hypoxemia, was happening more frequently in their Black patients than white patients. This did not go well. Senator Elizabeth Warren and Cory Booker were alerted to this, and they wrote a letter to the FDA and said, what is going on with pulse oximeters? You're supposed to be regulating these. The FDA picked up the phone and called me and said, how are we going to fix this problem? And I said, you've come to the right place. We know what to do about this. The gap in your regulation of pulse oximetry has been the fact that you have not required data from real clinical environments in the validation data that you use for pulse oximeters. You're taking data only from ideal lab conditions to regulate pulse oximeters. So, with the help of Carolyn Hendrickson and Mike Lipnick and others, we put together what's called the EQUIOX study. This was going to be the world's, we tried to design it so that it would really be innovative and really be the first accurate assessment of how pulse oximeters were doing in a real clinical environment. And really, Carolyn Hendrickson and her colleagues at San Francisco General, we owe them most of the credit for this. We had some ideas, but boy, Carolyn and her team really did the bulk of the work here. And these are data from her study that I'm going to present to you next. So the study objectives for EQUIOX were really to test whether pulse oximeters were performing differentially in our patients with darkly pigmented skin. I mean, there were so many problems in the retrospective analyses done by Schoeding and others. There was real questions about the timing of the blood samples. There was questions about how the blood analysis was even done. There were major questions about how they categorized race, and none of those studies really quantified skin pigment. And we knew that skin pigment, not race, was probably at the heart of the problem. So the EQUIOX study then was prospective. We enrolled adult ICU patients who had arterial lines at Zuckerberg San Francisco General Hospital, which of course is a level 1 trauma center, safety net hospital in our community. And the study was done between 2022 and 2024. And we had these direct observations between what the clinically used pulse oximeters were doing and arterial samples. So we quantified skin pigment in a very careful way. This had never been done before. We used a colorimeter device, which creates a representation of human skin pigment in multi-dimensions, produces a thing called the ITA angle, which I don't even completely understand to this day, but it's an objective assessment of the amount of melanin in a person's skin. It's reproducible by different observers and provides, as I said, an objective measure. We also used a scale devised by Harvard sociologist Ellis Monk. So the Monk skin tone scale, it does a much better job of encompassing the full range of human pigmentation than the scales that we had used previously. So this is some of our team. Oh, multiple places. We measured at the site where the pulse oximeter is applied. We measured it on the forehead. a chest, yes, multiple sites. And, it's produced thousands and thousands of observations. So these are some of our team members obtaining these data. All right, so we enrolled a total of 631 patients. Their average age was 62. They had a pretty diverse range of comorbidities. 32% had diabetes. We had patients with heart failure, hypertension. But by the time we got our study going in San Francisco, the COVID pandemic was starting to wane, and only 4% of our enrollees had COVID. 76% were on ventilators. A lot of them were on vasopressors, and some had low perfusion. And our ethnic distribution reflected very precisely what we had promised to the FDA based on what we knew about the demographics in our community. So San Francisco was an excellent place to capture a range of not only patient skin pigmentation, but race. All right, so overall, our findings were a little bit surprising. We found that overall, we actually had a negative bias. So the pulse oximeters were reading a little bit too low. But if you look at the distribution of all the data with these violin plots, some things really start to stand out. So this is a plot of the bias in the readings according to different skin pigmentation groups. And you can see right away that the distribution looks quite a bit different. So there's in our subjects who are more darkly pigmented, the bias tended to be positive. So although negative bias protects against hypoxemia, 20% of our readings had a positive bias, and that was associated with dark skin pigmentation and self-described Black or African-American racial background. So this was mirroring in a lot of ways what we had seen in the lab and published back in 2005. So we did some very sophisticated statistical analyses. We had some really fantastically talented statisticians with us. And you can see that there's a trend toward more positive bias across the skin pigment range. And this is the ITA number. So this is objective skin measurement, and this is just race. Okay. So another really remarkable finding, and I think a very powerful one, was that clinically, pulse oximeters do much worse than what you'd appreciate by looking at the data that's included in the package insert with a pulse oximeter. So the clinical performance, the performance of pulse oximeters in a real-world clinical environment was distinctly worse than what you're led to believe from the manufacturers. So this has major implications for regulatory science. So the RMS was close to 4%. So by that standard, these pulse oximeters would flunk. They wouldn't get passed. So this is a real problem because the FDA has never required real-world clinical data. This is very idealized lab data. There's no missed hypoxemia here under ideal lab conditions with healthy subjects, good perfusion, comfortable environment. Whereas what Schoeding saw in real patients was definitely different, a real world impact. There's a lot of ways you might say, oh, this difference looks really small. It's not really that much. But if you look at it on a population level, it actually is quite impactful. And these are some really nice modeling that Carolyn Hendrickson did with these data. And depending on the level of bias or random error, let's just focus on this. With 4% random error, the distribution of the data into ranges where you would have hypoxemia is around 11%, compared to if the oximeter is reading only 2% random error, it's much less frequent. So accuracy does make a difference in missing the diagnosis of hypoxemia. It can be quite impactful. So the take-home message from this, I think, is a really important one for clinicians to appreciate. Most clinicians, I don't think, even our most sophisticated anesthesiologists don't really appreciate that pulse oximeters are not absolutely accurate. We tend to treat the numbers as the gospel. right? But pulse oximeters are not thermometers. And in the real world, you have to understand that pulse oximeters maybe are giving you an estimate of the patient's oxygenation. If you really want to know, better get a blood sample, or at least be very skeptical about the pulse oximeter, what the pulse oximeter is telling you. Our work in the lab also has involved something called the Open Oximetry Project. It's our belief that there should be free and open information about the performance of different pulse oximeters. And largely through the efforts of Mike Lipnick, the Open Oximetry Project now lists performance data from about 50 pulse oximeters that have been evaluated in our lab. So this is open source, open data. We use an entity called PhysioNet to upload all our data so other researchers can look at all our data and use it for different kinds of analyses. I think it's pretty cool, part of what we do. So here's our Equiox project team. This was the one that was, this is the team that was involved with the clinical studies, just an enormous number of really dedicated people from across the US and even internationally. And the open oximetry team includes these individuals. You'll probably recognize some of your colleagues or maybe even yourself here. All right. I'd like to return to John in closing. When John became an anesthesiologist, the scientific foundations for our specialty scarcely existed. His life's work, more than anyone before him, laid and strengthened that foundation. His many contributions are not only woven into the foundation of our daily work, but form the cornerstone for the modern basis of respiratory physiology and critical care. This is an image of Hypnos that appeared in the preface of that book that I read in 1997. Hypnos is the Greek god and patron of sleep and anesthesia. John invoked Hypnos when he wrote just a beautiful preface to John Nunn's book. He wrote, "The lighted candle respires, and we call it flame. The body respires, and we call it life. Neither flame nor life are substance, but process. The flame is as different from the wick and wax as life from the body, as gravitation is from the falling apple, or love from a hormone." Newton taught science to have faith in process as well as substance, to compute, predict, and depend upon an irrational attraction. Science and knowledge weaves physiology with clinical medicine and forms the basis for our specialty. I just am continually blown away by that insight and the knowledge of how much John contributed to things that we do every day and how much he meant to so many of us. So thank you for letting me share that journey with him.
Well, wow, thank you, Phil. Just what a remarkable career. And I think I'm really thrilled to see so many of our residents here who didn't get a chance to interact with him, but to understand the history, the importance of John Severinghaus to our department, to our specialty, and those are completely interwoven. And it's why it's such an honor for me to be a part of this department. We've had many others that you saw, Ted Eager. I also want to announce that in October, we're still finalizing a date. We're going to have a celebration of the life and the career of Ron Miller, who passed away recently, and we will update you on that. But thank you all for coming. I think we have do we have snacks? Okay, thank you, Olivia. The snacks are outside, so snacks and socializing. Again, thank you all for coming, and thank you, Phil, for just a really remarkable lecture. Awesome. Thank you. Perfect.